This is a guide of how you can perform the ELISA test for the aCL kit. It will give you visually all the information you need to do the test, including the use of the Terra software. All TheraTest kit boxes are labeled with the content of the kit, the expiration date, and the kit's lot number. Allow all of the reagents and microplates to warm to room temperature, which is 18 to 25 degrees Celsius, by removing them from the kit box as demonstrated below. This will take at least 30 minutes. The kit contains a data sheet presenting the range of appropriate measurements that the calibrators and controls must fall within. The units and ranges change for each new lot. Prepare all necessary equipment to perform the test. They are listed below. Prepare the 1X aCL wash buffer. Using a graduated cylinder, add 900 milliliters of deionized water and 100 milliliters of 10X concentrated aCL wash buffer. Following the preparation of the 1X aCL wash buffer, take the label from the concentrated 10X aCL wash buffer and place it on the newly prepared buffer. Initial and label the buffer with the date it was created. Next, label a dilution tube for the diluent blank, positive control, negative control, and each patient specimen. Place the labeled tubes into the dilution block in the location indicated on the plate map. Please note that calibrators are pre-diluted, therefore the calibrators will not need to be diluted further. Add 1 milliliter of the aCL specimen diluent to the diluent blank, positive controls, negative control, and all patient specimens. Using a new tip each time, pipette 10 microliters of controls and patient specimens into their corresponding dilution tubes. Cap all samples firmly after adding specimens. Slowly invert the dilution tube rack 5 times in order to mix the samples. As a means of ensuring adequate mixture of the samples, watch as the fluid moves through the dilution tubes. Label 3 additional tubes representing the calibrators, IgM, IgG, and IgA, and place the tubes into 3 empty slots in row A. Pipette 250 microliters of each calibrator into the labeled tubes. The calibrators are pre-diluted and should not be diluted further. Using a pair of scissors, carefully loosen and remove the caps from each sample tube. On the left is an example of a TheraTest plate frame and wells prepared to run an aCL assay. On the right is a plate setup representing the sample layout for the aCL assay. Please note, calibrators, controls, and patient specimens are run in duplicate. Set a timer for 30 minutes. Using the multi-channel pipette, carefully draw up 100 microliters of calibrators IgM, IgG, and IgA. Observe each pipette tip and ensure the fluid levels are equal throughout. Load the calibrators into their corresponding wells in row A of the microwell plate. Using the multi-channel pipette and new pipette tips, carefully draw up 100 microliters of the positive controls. Load the positive controls into their corresponding wells in row C of the microwell plate. Using the multi-channel pipette, carefully draw up 100 microliters of the diluent blank, negative control, and remaining patient specimens as demonstrated below. Load the controls and patient specimens to their corresponding wells. Start the timer that was set for 30 minutes. When the 30-minute timer sounds, carefully flick the microwell plate into the sink in a swift downward motion. Make sure the strips of wells are firmly placed in the plate frame. This will serve as a precautionary measurement to prevent the wells from falling out of the frame and into the sink. Pour the previously prepared 1X aCL wash solution into a clean reservoir. Set the multi-step pipette to number 5. This will load 250 microliters with each press of a lever. Insert 8 new pipette tips firmly. With the tips in the wash buffer, depress the multi-step pipette plunger to load the wash solution. Carefully pipette 250 microliters of 1X wash into each column. Lay the pipette down on the bench with the tips facing down and out. This will prevent any fluid backing up into the pipette and contaminating it. Decant the wash into a sink. Repeat this wash procedure 2 additional times for each plate for a total of 3 washes. Firmly and carefully tap each plate upside down into a paper towel. This is performed to remove as much excess wash as possible. Pour the IgM, IgG, and IgA conjugates into new reagent reservoirs. Use new pipette tips to load the conjugate. Set a timer for 30 minutes. Starting with the IgM conjugate, load 100 microliters into column 1 and column 2. Please note, when more than 4 patient specimens are tested, IgM conjugate will be loaded into column 7 and column 8 as well. Refer to the plate layout for additional help. Next, load 100 microliters of the IgG conjugate into column 3 and column 4. Please note, when more than 4 patient specimens are tested, IgG conjugate will be loaded into column 9 and column 10 as well. Refer to the plate layout for additional help. Finally, load 100 microliters of the IgA conjugate into column 5 and column 6. Please note, when more than 4 patient specimens are tested, IgA conjugate will be loaded into column 11 and column 12 as well. Refer to the plate layout for additional help. This is an example of a microplate loaded with all 3 conjugates. The leftover conjugates may be poured back into the reagent bottle. Following the 30-minute incubation, wash the plate with the same technique as the first wash cycle. Wash each plate 3 times. Use a new reservoir for the addition of the chromogen. A full plate of wells will require 10 milliliters of chromogen added to a reagent reservoir. For 2 plates of wells, add 20 milliliters of chromogen. Plan accordingly, as excess chromogen may not be poured back into the original bottle. Use new pipette tips to load the chromogen. Set a timer for 15 minutes. Start the timer as you begin to load chromogen into the first column of wells. Do not wait to start the timer until the entire plate is loaded with chromogen. Draw up 100 microliters of the chromogen reagent into new pipette tips. Pipette 100 microliters of chromogen from a reagent reservoir into all of the wells. Try not to touch the edge of the wells with the pipette tips to avoid contamination. Never pour chromogen remaining in the reagent reservoir back into the reagent bottle. Simply discard any leftover chromogen. This is an example of wells containing chromogen following the designated 15-minute incubation period. Add stop reagent to the reservoir before the timer expires. Draw up 100 microliters of the stop reagent into new pipette tips. Add the 100 microliters of stop reagent to all of the wells. Tap the side of the plates gently to mix the samples in the wells. The rest of the stop reagent may be poured back into the reagent bottle. Below is an example of a completed aCL assay. The samples in each well are indicated. Place the developed plate into an ELISA reader for analysis. To read a new plate on the Terrace software, select File, Reading, New. Select aCL from the list of assays. Select Reader, Read and Collect. The reader will take 75 to 85 seconds to read the plate. This is an example of absorbance measurements displayed on Terrace software for the aCL assay. Cells A1 and A2 represent the IgM calibrator. Cells A3 and A4 represent the IgG calibrator. Cells A5 and A6 represent the IgA calibrator. Cells B1 through B6 represent the diluent blanks. Cells C1 through C6 represent the positive controls. Cells D1 through D6 represent the negative controls. The remaining cells represent the patient samples. After measuring the absorbance values, select the Sample IDs tab. Select Edit On. Enter the sample IDs from the assay in the same order the samples were loaded into the microplate wells. Select Save once all sample IDs have been entered. A new screen will pop up. Type in the name of the test that is run, followed by the date and initials of the individual who ran the test. Please see the example. Select OK to save the assay. This concludes the presentation provided by Theratest Laboratories in reference to performing the aCL ELISA assay, as well as a demonstration of test result storage within our proprietary software Terrace. If further clarification or assistance is required, feel free to contact our technical support department at 1-800-441-0771.